In this video, I explain the Parker hydrophilicity scale, a classic sequence-based method used to identify hydrophilic regions in proteins, and how to implement the method in Python step by step. What is the Parker hydrophilicity scale? It's a method used to measure how water-loving each amino acid is in a protein sequence. By scanning the sequence with a sliding window, it identifies hydrophilic regions that are likely exposed on the protein surface. These regions are important because they are potential antibody binding sites, also known as epitopes. How does it work? The Parker hydrophilicity scale scans a protein sequence using a sliding window. For each position, it calculates the average hydrophilicity of nearby amino acids. This creates a smooth profile along the sequence. Peaks in this profile indicate hydrophilic regions, which are likely exposed on the protein surface and may serve as potential antibody binding sites. Here is the Python implementation of Parker hydrophilicity scoring. This is Parker Scale Dictionary, which assigned a hydrophilicity score for each amino acid. The run_Parker function calculates Parker hydrophilicity scores for each amino acid using a sliding window. It takes protein sequence as input, and the default window size is 7. If you want to large or narrow the window, you can define a custom size here. For each position, it averages the Parker scores of nearby amino acids and returns a list of scores that indicate how hydrophilic each region is. The print_results function prints the hydrophilicity results in a clean table. It also adds biological meaning by labeling each residue as hydrophilic or hydrophobic based on a chosen threshold. This main block runs the program using a sample protein sequence. It calculates hydrophilicity with the Parker method and then prints the results in a readable format. After clicking Run, the result will come out. Why does this work for epitope prediction? Antibodies can only bind to regions that are exposed on the protein surface. These exposed regions are usually hydrophilic, meaning they interact well with water. The Parker hydrophilicity scale helps identify these regions by finding hydrophilic peaks along the sequence, making it a simple and effective way to predict potential epitopes. An important limitation is that hydrophilicity alone is not enough to accurately predict epitopes. Many real epitopes are discontinuous, meaning they depend on the 3D structure of the protein, not just the sequence. Also, not all hydrophilic regions are true binding sites. So, the Parker scale is useful as a first step, but it should be combined with structural or machine learning methods for better accuracy.